Hello everybody, and welcome to today's webinar, Optimize Your Flow Cytometry, Best Practice for Sample Preparation, Staining and Analysis. Your host will be Mike Blundell, one of our product managers here at ABD Ceratec. Mike graduated from the University of Edinburgh with a BSc in Immunology. He then moved to University College London, where he joined the group of Professor Adrian Thrasher and obtained his PhD. His thesis research was focused on the primary immunodeficiency Wiskott-Aldrich syndrome. He investigated novel activating mutations and developed lentiviral vectors for use in gene therapy treatments, some of which have now been used in clinical trials. Mike contributed to over 50 peer-reviewed papers and has now left academia to become a product manager for flow cytometry. As we are trying to make this webinar as interactive as possible, please submit questions or comments via the Q&A box located at the bottom of the screen throughout the webinar. We will try to answer as many questions as possible in the session following the actual presentation. Should any questions remain unanswered, we will mail you a reply. The webinar will be recorded and an on-demand version will be available shortly after the webinar on the ABD Ceratec website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide, which describes solutions for the most common technical problems. I hope you will enjoy the webinar, and now handing over to you, Mike.
Hello everybody, welcome to this flow cytometry webinar. I would like to give you a brief overview of what we are going to discuss during the webinar. Firstly, I will discuss sample preparation and the different methods and considerations you may need for different samples. After that, I will describe some protocols that are useful for surface, intracellular and nuclear staining, and give some examples of how you can improve your staining. I will then discuss how dead cell discrimination and then doublet exclusion can be important for the analysis of your cells. Finally, I will go through step by step how these methods can be incorporated into a 4-color antibody staining workflow. So firstly, I will talk to you about best practice for sample preparation, what you have to consider when harvesting cells from different tissues, and why careful sample preparation is essential to obtain viable cells, which will help you obtain high-level flow cytometry data. So what are the considerations when making single-cell suspensions? Firstly, what is your starting sample? You may be growing cells in culture, in which case you will have to take into account whether the cells are in suspension or adherent. Suspension cells often need only to be harvested and washed in PBS to remove contaminating proteins or cell debris. The cells can then be resuspended at the correct concentration in a FAC staining buffer ready to be stained. We recommend using PBS with 1% BSA as a staining buffer, although you may wish to use fetal calf serum. If you are using adherent cells, you will have to consider the method which is best to detach the cells from the flask. Commonly, trypsin is used to create single-cell suspensions when passaging cells. However, this can be damaging to the cells if left too long. This may not be the best method for some surface markers, as the trypsin can cleave epitopes, resulting in reduced or no staining. A common, more gentle method of creating a single-cell suspension can be to use acutase. This is used at a lower concentration than trypsin and preserves more epitopes. After detachment, the cells can be washed in PBS and resuspended at the right concentration. Some cells, such as primary macrophages and dendritic cells, are often harvested by scraping to try and be as gentle as possible. However, whatever method you choose, there will always be a loss of cells and dead cells present. In order to make a single-cell suspension from bone marrow, the bones need to be harvested with as little muscle attached as possible. The ends are removed and the bone marrow core can be flushed out with PBS or media using a needle and syringe. The optimal size of needle will vary depending on the species and age of the animal. The bone marrow plug can then be disaggregated into a single-cell suspension by passing through a fine gauge needle followed by being passed through a filter, and this will ensure there is no contamination such as muscle or pieces of bone. After disaggregation, the cells can be washed in PBS and resuspended at the right concentration. If you are looking at secondary lymphoid tissue, such as thymus, spleen, or lymph nodes, you will have to mechanically disaggregate the tissue. This is commonly done by homogenization of the tissue or mechanical disruption using the plunger from a syringe. Care has to be taken here, as being too aggressive can lead to many dead cells, whilst being too gentle may lead to ineffective cell recovery. More gentle techniques can be used, such as flushing out using a needle and syringe, but cell recovery may be reduced. After harvesting, the cells can be washed in PBS and resuspended at the right concentration. Finally, after you have made a single cell suspension, you can pass the cells through a filter to remove any remaining clumps or debris. You may wish to change the size of filter depending on what cells you are recovering. For example, lymphocytes can pass through a 40 micron filter, whereas dendritic cells may need to be filtered through a 70 micron filter due to their larger size. You may also be using samples which have been previously frozen. In this case, you will need to rapidly defrost the cells in a water bath at 37 degrees and ensure you remove the DMSO from the defrosted cells quickly by resuspending the cells in a large volume of cold PBS-BSA or PBS containing fetal car serum before centrifugation. The cells are ready to be stained now. A higher viability, however, may be obtained with some cells if they are left overnight, and some epitopes may need the cells to be in culture for a period of time to be expressed. Another popular sample used in flow cytometry is peripheral blood. Purification of peripheral blood can be very straightforward. First, though, you have to make sure you prepare the blood sample correctly. This starts with choosing the right anticoagulant. For most antibody staining, any of the common anticoagulants such as heparin, acid citrate or EDTA are fine to prevent blood clotting. However, for some techniques, such as intracellular cytokine detection and certain surface epitopes such as integrins, it is advisable not to use EDTA. If you're unsure, then you can check the datasheet of your antibody prior to blood collection. Red blood cells make up 95% of whole blood and can make detecting the leukocytes difficult during analysis. Therefore, removal of the red cells is an important step which can be done before or after staining with antibodies. If removing the red cells is not an option, you can use CD45 staining to gate out the red blood cells as they will be negative. One method to remove red cells is to use a density gradient such as Ficol or Lymphoprep. Here, the blood is diluted 1:1 with either PBS or media and slowly layered on top of the Ficol. This is then centrifuged to separate the different fractions. As we can see on the slide, a layer of cells called the buffy coat, consisting of mononuclear cells, are held at the interface between the plasma and the Ficol and can be removed by pipetting. The red blood cells pass through the Ficol due to their higher density. What should be noted is that the granulocytes will also pass through the Ficol layer, so this is not suitable if this is the cell population you're interested in. Platelets are found in the plasma layer. Samples which contain nucleated red blood cells, such as avian blood or fetal samples, will not be able to separate the red blood cells using this method due to the reduced density, as they have a nucleus. Pre-incubation with antibodies, which can aggregate red cells, may be needed to remove these cells. To ensure good cell recovery and distinct layers in your density gradient, the Ficol or Lymphoprep should be at room temperature, and most importantly, the centrifugation step should be performed without the break. Another method to remove red cells is to use hypertonic lysis. This method allows you to remove the red cells whilst preserving all the other populations in your sample. Methods include using water, ammonium chloride, or specialized red cell lysis solutions, such as erythrolyze. With all lysis methods, care has to be taken to ensure you get sufficient lysis of the red cells to enable you to easily distinguish the leukocytes without leaving the cells too long in the lysing solution. This will lead to an increase in cell death. Finally, with all sample preparation, the time taken between harvesting the sample and preparation for flow cytometry can have a large effect on the cell viability. Ideally, this time should be as short as possible to maintain the highest viability possible. If you have to store samples, then tissue should generally be stored at 4 degrees and whole blood at room temperature. Here is an example of the type of dot plot that can be obtained from whole blood that has been lysed to remove the erythrocytes. It is easy to distinguish between different cell populations based on the forward and side scatter. As can be seen on this plot, the lymphocytes, monocytes and granulocytes have been identified and gates placed around them. For some samples, however, specific cell populations may not be readily identifiable. What is also visible on this plot is debris and non-lyse red cells present in the lower left corner. This can be removed during acquisition by increasing the threshold of detection or during analysis using a gating strategy. In addition to mechanical disruption or homogenization, extraction of certain cells from secondary lymphoid tissue and cells from solid organs can require enzymatic digestion. The enzymes are required to break down the extracellular matrix and are often tissue-specific. Without use of the enzyme, you may not obtain a single-cell suspension, or you may not release your cells of interest. This will lead to an underestimation of your cell numbers. A couple of examples of where enzymes are required are F480 macrophages and follicular dendritic cells. Although F480 staining is really strong on spleen sections, red pulp macrophages are not readily released with physical treatment alone. Collagenase treatment, which cleaves peptide bonds in collagen, is required to release these cells. Follicular DCs are only released from germinal centres with treatment with liberase and DNase 1. An example of how mechanical action and enzyme digestion can be seen in the following facts plots. Nakashima et al. Were interested in Kupffer cells in murine models of cholesterol disease. They minced the liver with scissors, followed by incubation with 0.5 mg per ml of collagenase at 37 degrees for 15 minutes. The resulting cell suspension was then filtered through a steel mesh prior to staining with F480, CD68 and CD11B, and this identified different populations of Kupffer cells. The plots show clear differences between control and disease models for both CD68 and CD11B F480 double positive cells. An important consideration with sample preparation is the number of cells. Firstly, how many cells you're expecting within your population. Are the cells an abundant cell type? If you're looking at rare populations, will you have enough starting material to collect enough cells to be statistically significant? As you can see from the list of the cells present in human blood, if you're investigating granulocytes or T cells, there'll be a plentiful supply of cells, whereas NK cells and dendritic cells are at a much lower frequency. This has a large effect on the number of cells you need to acquire in your experiment. Examples of the number of cells collected based on cell frequency is shown in this table. Based on a starting population of 1 million cells and a frequency of 10%, if you acquire all the cells, you'll have 100,000 cells. However, if you have a frequency of 0.1%, the number dramatically reduces to only 1,000 cells. If this frequency is based upon one marker, and you're starting to look at subpopulations and several markers, you have to ensure you start with enough cells and acquire enough cells to have a statistically significant positive population. Because flow cytometry relies upon a stream of single particles, or in this case cells, to pass through the laser, it's very important you avoid cell clumps as much as possible. Cell clumps can block the nozzle of your cytometer. To try and prevent clumping, it is advisable to have all your media and wash buffers at 4 degrees and keep the cells on ice as much as possible. This will help slow down cell metabolism and reduce the amount of necrosis and apoptosis. Another method of preventing cell clumping is to include DNase 1 or EDTA in your resuspension buffer. When cells die, they release DNA, which is sticky and therefore promotes cell clumping. The inclusion of 25 to 50 micrograms per ml of DNase 1 will help remove the DNA. The inclusion of EDTA at concentrations of up to 5 millimolar will help to prevent cation-dependent cell-cell adhesion, as we're using cation-free media. Finally, prior to acquisition, passing the sample through a 70-micron filter can help to remove clumps. When resuspending your cells, there are a few simple things that can be done to help prevent excessive cell death. Firstly, avoid vortexing, as this will damage fragile cells. Avoid excessive centrifugation and sediment at the lowest speed you can. Don't aspirate off all your media from your cell pellet, as cells will die in a dry pellet. Avoid creating bubbles as the surface tension may damage your cells. Finally, count and resuspend your cells at a concentration of 1 to 10 times 10 to the 6. This can be done with a hemocytometer, or you could use an automated cell counter such as a TC20, which can provide you with an accurate cell count in 30 seconds. So, why is cell health and viability so important? Well, in flow cytometry, the quality of your data will be determined by the quality of your sample. The quality is determined by the viability and yield of your cells. A high number of dead cells, as we have already discussed, can lead to clumping of your sample, making it no longer a single cell suspension. And a high number of dead cells can also result in high autofluorescence and can lead to an increased amount of non-specific antibody binding. As can be seen in these cartoons of typical antibody staining, we have a negative population coloured in black and a positive population coloured green. On the left, if you had good staining with little background, you will easily be able to identify your positive cells. We can see, as we move through the images to the right, as the background increases, the positive cells become less defined, until there is an overlap between the negative and positive population. This is seen here in the red circle. And this will result in an inability to resolve weakly positive cells or rare cell populations that are found within this red circle, and can lead to difficulty with gating strategies and inaccuracies in your results. And this difficulty increases as you add increasing numbers of fluorophores to your staining panel. If you're interested in more detailed step-by-step protocols for cell preparation for flow cytometry, then we have protocols on the website, also in PDF form which you can download. Just go to the Application Resources section of the ABD website, and there you'll find protocols for flow cytometry. So the next thing I want to discuss is some basic staining considerations when using antibodies to identify surface, intracellular and nuclear epitopes. There are some basic considerations for all staining. Firstly, stain your sample in a small volume to avoid excessive dilution of your antibody. A good place to start would be to stain 1 million cells in a volume of 100 microliters, although you need to optimize this for your own particular cell type. Next, if possible, stain your sample on ice. Keeping your cells on ice slows down death and the cell metabolism, maintaining the cell viability, but it can also prevent internalization of your antibody. Some antibodies are temperature sensitive, and the binding is not optimal at 4 degrees, so check the datasheet if you're unsure. Also, some cells and some experiments require the cells to be maintained at room temperature or 37 degrees, so check your experimental protocol. One thing to remember is that antibody binding is a dynamic event, and the time required for staining may need to be increased at 4 degrees to promote the on stage rather than the off stage. Finally, I would suggest performing all staining with antibodies that are fluorophores in the dark or out of direct sunlight to avoid photobleaching. In flow cytometry, there are 3 basic types of staining. There is cell surface staining, which is the simplest and generally the easiest to perform when using directly conjugated antibodies. This is due to the availability of specific antibodies and access to the epitope. Then there is intracellular staining, and this can be more difficult. Intracellular staining requires you to fix and permeabilize the cells to obtain access to the epitope. As there are a lot more proteins inside the cell, the amount of non-specific binding can be increased. You are also often required to use indirect staining where unconjugated primary antibodies are detected with conjugated secondaries due to the lack of antibodies available. Whilst this may have the benefit of amplifying your signal, it will also amplify the background. The number of markers detected at the same time is diminished when using indirect labeling, as the primaries cannot be of the same subclass or species to avoid the secondaries binding to multiple targets. Finally, there is nuclear staining. This can be useful to determine cell cycle as well as nuclear localized epitopes. Nuclear staining, like intracellular staining, also requires fixation and permeabilization. In addition to permeabilization of the cell membrane, the nuclear membrane also has to be permeabilized for access to the dyes and antibody. For all cell staining with antibodies, it is important that the cells are in the right buffer. To minimize protein-protein interactions due to hydrophilic-hydrophobic binding or charge, it is advisable to include BSA or FCS in your staining buffer. Cells are resuspended in the staining buffer, and then antibodies are added at the recommended concentration, followed by incubation for about 30 minutes. The cells are washed to remove excess antibody and can be analyzed straight away or fixed for analysis at a later date. However, some cells, such as NK cells, B cells, dendritic cells and macrophages, have Fc gamma receptors on their cell surface. Fc gamma receptors allow antibodies to bind via the constant region of the antibody, leading to increased background staining. Pre-incubation with an Fc blocking antibody for about 10 to 15 minutes will prevent binding of other antibodies via the Fc portion. You can then continue staining as previously described, making sure you don't wash off the Fc block to ensure you get specific staining. As can be seen in this overlaid histogram, THP1 cells, a human monocytic cell line, which has Fc gamma receptors, was stained with CD11A FITC. Without an Fc block, both the negative isotype control, which is the black line, and stained cells, the filled blue histogram, are positive for CD11A. However, as can be seen on this overlay below, when an Fc block is added, there is a clear difference between the isotype control and the positive cells. Intracellular staining has more steps than surface staining. Cells should be resuspended in PBS-BSA, and if required, cell surface antigens can be stained as previously described. The cells are then fixed in an appropriate fixative for 10 to 20 minutes, washed and then permeabilized to allow access of your antibody. The directly conjugated antibody can then be added for 30 minutes, avoiding direct sunlight. The excess antibody is then washed off and the cells can be analyzed. Alternatively, if there is no directly conjugated antibody available, a fluorescently labeled secondary antibody can be introduced as an additional step. As I've previously mentioned, intracellular staining requires you to fix and then permeabilize your cells. Therefore, fixation is an important first step. Choosing your fixative can be important. Of the fixatives available, one of the most common is formaldehyde. Formaldehyde creates bonds between lysine residues, resulting in cross-linked proteins. It is usually used at concentrations between 0.5 and 4% in PBS, depending on your sample. For example, fluorescent proteins often require higher percentages of fixative. When samples are fixed, they should be stable for 1 to 2 weeks if kept at 4 degrees. However, if they're kept for a long period of time at 4 degrees, you should remove them from the fixative after the initial fixation period and not left for more than 2 to 3 hours before being stored in PBS. An alternative method of fixation is alcohol. Typically, cells are fixed in 7% ethanol or methanol. When alcohol is chilled to minus 20 degrees and added to cells, they are fixed by denaturing and precipitation of proteins. One of the benefits of using alcohol is that, as a fixative, is that it dissolves lipids at the same time. Therefore, you do not need to include a separate permeabilization step. An additional benefit of fixation in alcohol is that the samples will be stable for long periods of time when stored in the alcohol at either 4 degrees or at minus 20. However, as the proteins are fixed by coagulation, the epitope may be hidden when alcohols are used. After fixation, the next step for intracellular staining is permeabilization. Choose the most appropriate permeabilization method possible. The most common permeabilization reagents are detergents. However, as I've already mentioned, alcohols will permeabilize and fix. The choice of detergent is important, as insufficient permeabilization can lead to no staining. Alternatively, there may be high levels of background if unbound antibody cannot be washed out of the cell sufficiently. Weak detergents such as digitoning and saponin will permeabilize the cell membrane. However, if you want to include the permeabilization of the nuclear membrane, you will need to use a more harsh detergent such as Triton X100. When choosing your permeabilization reagent, there are a few things to consider. Some fluorophores attached to the antibodies are large, for example PE and APC, and may require many washes to remove excess. Alternatively, your protein target may be small and could leak out of the cell. To help you with your intracellular staining, I've listed here a few staining tips which may be required to help you optimize your staining. Firstly, some epitopes and fluorophores may be fixation sensitive, and therefore the choice of fixative may be important. PE and APC are large proteins and therefore can be affected by alcohol fixation. The order of the stain and fixation is important. Stain surface antigens before you look at intracellular antigens, as the permeabilization can affect surface staining. Fixation can be done before or after surface staining, depending on the sensitivity of the surface epitope. There are commercial reagents available, such as leucoperm, which contain fixation and permeabilization buffers. They are convenient and have been optimized to be used with most commercial monoclonal antibodies without altering the cell morphology. Some detergents, for example saponin, may need to be included throughout the staining protocol. including during the washes, and this will ensure you have efficient washing off of excess antibody. Always use your fixatives cold. Fixation is an exothermic reaction, and this could affect your cells. Fixation may alter your cell size and the level of fluorescence, particularly with tandem dyes. Therefore, you may need to alter the forward and side settings afterwards when you acquire your sample. And if you are comparing expression levels between samples, it's best to treat them all in the same manner to allow comparison. When adding your fixative, gentle mixing will help to prevent clumps, ensuring you have a single cell suspension. Finally, if possible, avoid using biotin and the fluorophore FITC in intracellular panels. There is endogenous biotin in some cells which will need to be blocked with unconjugated streptavidin to prevent high background. FITC has also been shown to bind non-specifically through electrostatic interactions to certain cell structures, again increasing the level of non-specific staining. Nuclear staining also has specific staining protocols. When staining DNA, fixation in alcohol is generally the preferred method, as it results in better profiles. After fixation for at least 30 minutes on ice, the cells can be washed and resuspended in PBS. The DNA can then be stained using common DNA dyes such as propylium iodide. However, as propylium iodide also stains RNA, an incubation step with 100 micrograms per mil of RNase A for 30 minutes at room temperature is required before staining to remove the RNA. Alcohol fixation is not compatible with fluorescent proteins and some surface staining. If you require to examine fluorescent proteins or surface markers as well as examining the cell cycle, it may be better to fix using formaldehyde followed by alcohol resuspension. This will, however, result in poorer DNA profiles, so some optimization will still be required. In addition, some protocols for nuclear staining may require the use of permeabilization reagents in conjunction with methanol after fixation to obtain optimal staining. For those that are interested, there are detailed protocols on the ABD Serotech website that give more details on surface and intracellular staining. Just go to the web link here for a list of flow cytometry specific protocols, and these are also available as PDFs to download. Now I would like to move on from staining to other ways in which you can optimize your flow cytometry. First of these is dead cell discrimination. So why is dead cell removal important, and what are the best ways to do it? Well, dead cell removal is important because, as we mentioned earlier, dead cells have increased autofluorescence and have increased binding of antibodies through non-specific interactions. The result of this is that the negative population will be more positive and the dynamic range of staining will be reduced, resulting in weakly positive cells and rare cells not being able to be identified from the negative population. Although dead cells often have a different forward and side scatter profile compared to live cells, and this method is often used to distinguish between live and dead cells, this is not always the case. Because of this, dyes have been developed to distinguish between live and dead cells. And one group of dyes are the nucleic acid binding dyes. Examples of these include propylium iodide and 7-AAD, which are excitable by both the 488 nanometer laser and the 561 nanometer. When they bind to double-stranded nucleic acid, they fluoresce. They are excluded by live cells as they are not membrane permeable and can be added directly to samples after being stained with antibodies. After addition of the dyes, the cells are briefly incubated and then acquired as normal. The dead cells can be identified and removed from the final analysis, as they will be fluorescent. As these dyes rely on membrane integrity, it is not possible to fix these samples. An example of how the presence of dead cells can affect staining is shown in this slide. J772 macrophages were removed from culture and stained with the myeloid marker CD11b and the T-cell marker CD3. Cells were selected based on their forward and side scatter profile and the fluorescence determined. As can be seen in the fluorescence dot plot, there appears to be two distinct populations, one positive for CD11b and one positive for CD3. When we add propylium iodide to the stain, we can observe a live and dead population based on the level of PI fluorescence. The dead cells can now be excluded from the analysis and the fluorescence dot plot re-examined. After dead cell discrimination, the CD3 positive T cell population disappears, leaving just the expected myeloid CD11b positive stain cells. There are a second group of dyes available to enable you to discriminate dead cells from your samples, and these are the protein binding dyes. As can be seen in the image, these dyes will bind to both live and dead cells. However, when a cell has a compromised membrane, as seen in dead and dying cells, there is access to a greater amount of protein inside the cell. Therefore, they have a higher fluorescence. As can be seen in this dot plot, where heat-killed jerkats have been spiked into a live population and then stained with one of these protein dyes, Vivafix, there is a measurable difference between live and dead cells. The added benefit of these dyes is that once stained, the cells can be fixed without any reduction in the resolution between the live and the dead cells, and this is seen here in this fixed sample. In addition to the added benefit of fixing your sample to now allow analysis at your convenience, they are available in a range of excitation and emission spectra for convenient addition to multicolour flow cytometry panels. So the last tip today to improve your flow cytometry is to perform doublet exclusion on your sample. Doublet exclusion, as the name suggests, is to ensure you count single cells and exclude doublets from your analysis. As a particle, in this case a cell, passes through the laser, it creates a pulse. A non-fluorescent cell will produce a pulse that is fluorescence low or negative, as is shown in black, whereas a fluorescent cell, shown in green, will produce a positive pulse. Unfortunately, doublets create a pulse which the cytometer cannot distinguish between those created by single cells. As can be seen in the diagram, a doublet containing a positive and a negative cell will produce a positive pulse. Furthermore, in doublets where there are different cells stained with different antigens, they will appear double positive when they are in fact two singly stained cells. Excluding doublets, therefore, is important to avoid false positives and skewing of your data. If you are sorting cells, excluding doublets is important, as this will lead to sorting of negative cells as well as positive cells. So how do you do doublet exclusion? Well, doublet exclusion is performed by plotting the height or width against the area for forward scatter or side scatter. Doublets will have double the area and width values of single cells, whilst the height is roughly the same. Now, one area where removal of doublets is also important is in cell cycle analysis. It is important to distinguish between single cells that have double the amount of DNA and doublets, as they will both show increased fluorescence when stained with a DNA dye such as propidium iodide. Fortunately, using the height or width versus area allows you to separate the doublets from the single cells containing 4n amounts of DNA. Cells containing 4n amounts of DNA have double the height and area values, whilst the width is roughly the same as cells containing 2n amounts of DNA. Therefore, disproportions between height, width, and area can be used to identify doublets. As can be seen in the dot plot, the doublets, here circled in red, are able to be identified by their increased area compared to single cells and excluded, ensuring only single cells are analysed. So you may ask, how can I check my cells before analysis? The simple answer to this is to check your cells down a microscope. A visual check of your cells can be useful at any stage of a flow cytometry workflow. It can be done prior to staining to check the general health status of your cells, and it can check the level of debris compared to intact cells, and to look for any unusual morphology in your cells. This will enable you to increase the threshold to remove excess debris, alter forward side scatter voltages to accommodate cell changes, or even abandon the staining altogether if the viability is poor. After staining, you can check the levels of fluorescence. This will depend on the fluorophore used and the filters available on your microscope or imaging system, such as the Zoe fluorescence cell imager shown here. A check of the health status of your samples and the number of cells to ensure the staining protocol has not adversely affected your cells can also be important. You can then determine whether it is worth acquiring your sample. Finally, after sorting, it is advisable to check your cells' health, if you're culturing them, and fluorescence levels to ensure you have the right population to proceed with other experiments. So finally, I would like to quickly show you where some of what I've talked about today fits into a typical flow cytometry protocol by showing you a basic immunophenotyping experiment using 4 colours. So whole blood was collected in heparin, but for this experiment any anticoagulant would be fine. And then the blood was aliquoted into 100 microliter aliquots ready for staining. The samples were pre-incubated with human seroblock to prevent binding to the Fc gamma receptors. The samples then had directly conjugated antibodies against CD3 for T cells, CD20 for B cells, and CD14 for monocytes, added at the recommended dilutions. The appropriate isotype controls were added to determine background, and single stain controls were done to allow compensation to be performed. The samples were then incubated on ice for 30 minutes, avoiding direct sunlight. After 30 minutes, the sample was lysed using ammonium chloride and washed in PBS-BSA twice before resuspending in PBS. At this point, 1 to 2 drops of propidium iodide was added for dead cell discrimination. After a further 10 minutes, the sample was acquired and 200,000 cells collected. As we can see from the forward and side scatter profile on this dot plot, we are able to see 3 distinct cell populations. However, there is also some debris seen at the bottom left, so we can put a gate around the larger cells to exclude the debris. Using the propidium iodide staining, we can gate on the negative and therefore live population. In this case, there are only a few dead cells, which is expected because this is a fresh blood sample. Using the height versus area from the side scatter, we are also able to perform doublet exclusion. As expected with fresh whole blood, there were few doublets, as shown in the red ellipse. Finally, the lymphocytes and monocytes were marked with regions to enable us to distinguish the staining in different populations. The lymphocytes are marked with a blue region, and the monocytes with a red region. The cells in the lymphocyte gate are coloured blue, and the cells in the monocyte gate are coloured red. As we can see in panel A, there is a clear CD3 positive population and a clear CD20 positive population within the lymphocyte gate, representing T cells and B cells, as expected. And within the monocyte gate, which are red, they are negative for both markers. In panel B, in the middle, this shows the CD3 positive T cells and the CD14 positive monocytes, with a negative lymphocyte population, which is the B cells. And finally, in panel C, we can see the CD20 positive B cells and the CD14 positive monocytes, with the negative lymphocyte population being the T cells. These stains are all mutually exclusive in peripheral blood, and as expected, there are no double positive cells, showing we have specific staining. I have not had the time to explain all aspects of flow cytometry today. However, this is a first in a series of planned webinars, and we will discuss other important topics such as fluorescence, controls, experimental design, and multicolour flow cytometry later in the year. In the meantime, if you are interested in further explanation of the principles of flow cytometry, then we have a flow cytometry basics guide available on our website as a PDF, where you can request a printed copy. In it is information on principles of flow cytometry, principles of fluorescence, data analysis, common protocols, troubleshooting, and some further reading. In addition to our popular flow guide, we have a wealth of flow cytometry resources on our website, including flow cytometry protocols, tips and tricks for generation of multicolour panels, intracellular flow cytometry tips, and fluorophore tables with excitation and emission data with relative brightness. All of these resources, as well as the flow cytometry guide, are also accessible from the resource list widget on the top right hand corner of your screen. So in summary, today I've shown you the importance of preparing your cells with care from various sources. I've also explained some of the basic principles of antibody staining for cell surface, intracellular and nuclear staining, with some tips that will help you optimize your staining. I have then gone on to show how dead cell discrimination and doublet exclusion are important aspects to be included in your workflow, and how they will improve your flow cytometry. Finally, I've shown you an example of a 4-colour immunophenotyping workflow where all these principles have been incorporated, resulting in bright, clear, specific staining of different blood cell populations.
Thank you, Mike, for that interesting and informative presentation. The Q&A session will begin shortly. Just in case you were not able to stay for the duration of the Q&A or missed your chance to submit a question, you can contact us via the ABD Ceratec website at abdceratec.com. Mike will be joined for this Q&A session by our colleague Steve Clasper. Steve has a PhD in biochemistry from the University of Liverpool and spent 16 years doing postdoctoral research between Liverpool and Oxford University. Steve has worked here for almost 10 years in research and development. And now over to you, Mike and Steve.
So thank you for that. We have some questions that have come through. And the first question we have is, there are times when I recover red cells from the interphase of a phycol. What is wrong? Well, quite often when you have red cells at the interphase, there's a few reasons why this can happen. First of all, make sure the brake is off when you centrifuge. You have to make sure you have a good separation of your layers. So as you're layering your blood onto the phycol, make sure that they do not intermingle. You should ensure that the phycol is at a room temperature. And if you still have red cells after you do all these methods, you can still lyse them using either ammonium chloride or commercial reagents such as erythrolyse. Or you can actually gate them out during analysis. So over to the next question. I want to do intracellular cytokine staining. Are there any specific steps I need to include in addition to the basic intracellular staining protocol? Well, for specialized intracellular staining, such as cytokine or chemokine staining, there's a few extra steps and precautions that have to be taken. When you are using blood as a sample, you have to make sure you collect the blood in the appropriate anticoagulant. EDTA is not a suitable anticoagulant when looking at intracellular cytokines. And there are also some recommended controls you should do. For example, you should include a control sample treated as your experimental or disease model if required. You should also have an untreated or an unstimulated sample and a fully stimulated sample. And this will enable you to determine the full range of expression of your cytokines. As cytokines and chemokines are secreted targets, they'll need to be trapped within the cell. And to do this, you'll use something like monensin or profeldin A. And these work by interfering with Golgi transport or the redistribution of proteins from the Golgi to the endoplasmic reticulum. So this holds the cytokines in the cell. And which one you need to use will depend on your species and which of the cytokines or chemokines you are looking at. Okay, the next question. What is the best way to prepare platelets for staining? Again, the best first thing to do is collect the blood in the right anticoagulant. This will often vary depending on what experiments you're performing. But I would recommend you using acid citrate dextrose. And platelets are very easily activated, so you need to mix the blood and the anticoagulant very gently. If you shake or pipette too much, you will activate your platelets and they can clump. I would recommend centrifuging the blood at 200 G for 20 minutes without the brake at room temperature. And after the centrifugation, you will see 3 layers in your tube. The bottom layer will contain the red cells. There'll be a thin layer of cells on the top of the red cell layer, and this will be the buffy coat. And sat above this, in a sort of a yellowish plasma, and this is the plasma which contains the platelets. You can transfer this top platelet-rich plasma layer to a new tube and dilute 1:1 with room temperature heaps. And you have to pellet the centrifuge now by centrifugation at 800 G for 15 minutes at room temperature. And again, do this without the brake. If you are wanting to avoid activating your platelets, it's advisable to include prostaglandin in this buffer. But if you're not worried about activating your platelets, you don't need to include this. After this centrifugation, you can wash your the platelets again in the heaps buffer, and then it's best to resuspend in Tyrode's buffer. and you resuspend them at the concentration you want to do your experiment in, or you're staining it. Remember that platelets, however, are very small, and when you're acquiring them, it's best to use the forward and side scatter on the log scale so that you can actually distinguish them from cell debris. Right, I'll pass over to Steve, who's got answers for a few more questions.
Okay, so my first question is, how fast should I spin my cells when washing? So when you're centrifuging your cells, you're trying to find a balance between forming a nice firm pellet but preventing loss of viability. And what you'll find is that spinning between 300 and 400 Gs will usually be best for this. You'll have a pellet which isn't too loose so it doesn't fall out when you decant, but also you should maintain the maximum viability. The next question is, can I use frozen cells and how should I prepare them? Often you need to use frozen cells from a tissue if you can't do all of your analysis in one go, and it's quite feasible to do this. What you need to do is harvest the cells from the tissue with your usual method, whether it's mechanical or some kind of enzymatic digestion. Then you need to form this single cell suspension, freeze it down in a standard freezing medium, preferably an aliquot that you would need for a single flow run. When you're ready to stain, you defrost them, use your normal staining protocol. But what you'll find is there'll probably be quite a high number of dead cells, so you'll need to do a live dead stain to improve the quality of your data. We also have another question on dead cells, which is, how do I remove them from my sample? Well, the easiest way to remove them from your analysis is to do a live dead gating with a viability dye. However, if you actually want to physically remove them from your sample, the best thing to do is a Ficol centrifugation as you do in a blood prep. What you'll find here is that the dead cells will actually pellet at the bottom, whereas on the interface between the media and the Ficol, you will find the live cells. You can then remove these carefully and do your standard staining. Another question on viability is, will a viability dye exclude apoptotic cells? Most viability dyes rely on a permeabilization of the membrane during cell death, which allows the dye to enter and bind to the nucleus. So cells in the early stage of apoptosis will have an intact membrane which will prevent the viability dye entering. However, it's easy to identify these cells using a material such as an Exin-5. The apoptotic cells will be positive for an Exin-5 but won't show any viability dye inside. As they progress through apoptosis, they'll actually start to allow the viability dye inside, and you can see them appearing. Final question I have here is about pulmonary macrophages, harvesting them from tissue plastic. They like to know whether these can be removed by scraping because they're quite adherent, and if this will cause a loss of viability and increase debris. Yes, scraping will usually result in these kinds of problems. What I would recommend is using cold PBS containing EDTA. If you pour this onto the cells after rinsing with standard PBS, let them sit in the cold for 5 or 10 minutes, you'll find that the cells will round up and can actually be removed a lot easier, either by gentle scraping or tapping. Another way to do this is with a gentle cell removing agent such as acutase, which will actually remove them and not cause a lot of cell debris. I'm going to hand you back to Mike now. I think he has another question.
Yes, so we have quite a few questions. So let's see if we can go through these. So we have a question that says, what is the recommending spinning time for pelleting? Well, this will depend upon your sample, and but generally, for cell population, 5 minutes at 300 to 400 G would be a good place to start. And also, we've asked, will washing and spinning remove your cell debris? Well, this will remove some of your cell debris, but it may be best to, if you have a lot of cell debris, to either phycol your sample again, or you can gate out the cell debris using the threshold on the forward scatter, and so that will help you to resolve your actual cell population. And I got a question here: is leaving your cells thawed overnight really encouraged for frozen samples? Because certain activation markers can change. Well, leaving your frozen samples overnight is only good for some samples, so you will have to change your protocol depending upon your cells. So if you're worried about your surface markers changing, then I would not recommend leaving frozen samples overnight. I would stain them as quickly as possible, and if it's all possible, I would actually stain fresh cells, because you will have a much better viability with your fresh cells. I have another question here. Is it compulsory to use isotype controls? Well, the short answer to that is no. However, you may wish to include them in your protocol. Isotype controls can be quite a controversial subject in flow cytometry, and we will be discussing isotype controls later on in the year in another webinar. However, the best advice at the moment I would give you about isotype controls is, if you're going to use them, make sure you're using them properly. Isotype controls are designed to determine the level of non-specific background staining, and they are not supposed to be used as gating controls. I've got a question here. Can you use FITSI for surface staining if you then intend to do intrastaining afterwards? And the answer to that is yes. FITSI is very good for surface staining. However, I would not, as I mentioned in the webinar, I would not use this for intracellular staining. You may get some reduction of your fluorescence when you fix, but as long as your controls, you have the right controls there, you should be able to account for this. Let's go through. We've got quite a lot of questions here. Okay, how many cells would you recommend to analyze when looking for a cell population of around 1%? Well, this will depend on how much sample you have. I would recommend that you run as many cells as you can. And if you want to have a significant number of cells, I would recommend to run about 1 million. And this will allow you to have 10,000 cells within your positive gate. However, it's not always possible to run that many cells, and it could take quite a long time. So it also will depend on how bright your staining is. And see if we go through more. Some more questions. There's quite a few here. This is going through. There's here, PE and APC are large molecules. Does that mean transcription factors should be stained with smaller fluorophores? Well, there's no hard and fast rule as to which fluorophores you should use, but I would suggest that as APC and PE are large, I would not use them in intracellular staining. Smaller dyes, such as the Alexa dyes, would probably be optimal. However, you may you may have to optimize this staining yourself, and it will depend on what antibodies are available. If there are no antibodies available with the fluorophores you wish, you will have to either find the best secondary or use PE and APC. Okay. I have one final question, and this is: when is it necessary to do fluorescence -1? controls? Well, fluorescence -1 controls are important when you're using multiple color panels for flow cytometry. I would recommend using fluorescence -1 when you're using any more than 4 or 5 colors, and this will allow you to gate to show your fluorescence spread. I will go into controls such as fluorescence -1 and isotype controls in another seminar later in the year. Okay, so that concludes the question and answer session for today, and I'd like to thank you for listening to this webinar. And any remaining questions which we've not been able to answer live, we will get back to you in the next couple of days via email, and we will endeavour to answer all your questions. Thank you.